Imagine you are walking into a movie theater.
Okay, I am with you.
You sit down, the lights dim, the screen lights up, and you see exactly one frame of a film.
Just one single frame.
Literally just one split-second image. Let's say it's a picture of a car suspended in midair right over a cliff. And then, boom, the lights come back up.
Well, that is a very short movie.
Right. And then someone walks in and asks you to explain the entire plot of the movie, the deep motivations of the characters, and exactly how the car ended up flying over that cliff.
I mean, I would look at them like they were completely crazy.
Exactly. You can't. Welcome to the Deep Dive. Today, our mission is to explore how scientists are finally, and I mean finally, building a molecular video camera. Because for decades, the microscopic world of cellular biology has been staring at that exact same single frame of a movie.
Yeah, they've been desperately trying to guess the plot of incredibly complex diseases from a single snapshot.
And we are unpacking a really fascinating source today. It's a highly intriguing 2026 Spotlight article from Molecular Cell, authored by Jasmine T. Plummer at St. Jude Children's Research Hospital.
Yeah, and we're also tying in some foundational research from Science magazine.
Right, because this is all pointing to a groundbreaking new technology called TimeVault. They literally call it a molecular time machine for single cells. So, okay, let's unpack this. Because to understand why a cellular time machine is so revolutionary, we have to look at why time was essentially broken in cellular biology to begin with.
Right, and that limitation fundamentally changes how we understand, the building blocks of life.
Yeah.
Because the plot of a living organism is completely dynamic, it's always moving, but the tools we've historically used to watch that plot unfold have been completely static.
We are essentially trying to understand a moving target using a still photograph.
Exactly.
So I want to break down why we've been stuck in this static era. It all comes down to how we read a cell's internal instructions.
Yes, the transcriptome.
Right, the existing single-cell transcriptome. So if you've ever looked into genetics, you probably know that DNA is the master blueprint. It's locked in the cell's nucleus.
A giant reference library that never leaves the building.
But the DNA doesn't actually build anything itself. It sends out photocopies of specific pages.
Right, and those photocopies are called mRNA. Messenger RNA.
And the complete collection of all those mRNA photocopies floating around inside a cell at any given millisecond, that is the transcriptome.
And reading that transcriptome is the absolute ultimate biological intelligence gathering. I mean, if you can read a cell's mRNA, you know exactly what that cell is trying to do right at that very second.
Right.
Is it trying to divide? Is it actively fighting off a virus? Or is it mutating into a cancer cell? The mRNA tells you the immediate biological priority.
But capturing those instructions creates this massive mechanical problem.
Yeah.
In the source diagram, there's literally a camera icon next to the phrase gene expression from a snapshot in time.
Yeah, they use that camera icon for a reason.
Because to read the mRNA, scientists use something called a barcode UMI. This stands for Unique Molecular Identifier.
Exactly, a UMI.
It functions like a microscopic tracking number, like a tiny molecular barcode you'd scan at a grocery store. It allows researchers to quantify exactly how many copies of a specific instruction a cell was making.
But here's the catch, and it's a huge catch. To attach those barcodes, the researchers have to literally burst the cell wide open.
Wait, they have to destroy it?
Yes. They have to perform cell lysis because the sequencing chemicals and the reagents required to attach those UMIs, they are just way too bulky and harsh to peacefully pass through a living cell membrane.
So they just blow the doors off.
They dissolve the cell's protective outer lipid layer. You explode the cell to get the mRNA out or to get the tagging chemicals in.
Which means the exact moment you take the measurement, you kill the subject.
You kill the subject every time.
It is like trying to figure out how a mechanical clock works by just smashing it with a hammer and looking at the scattered gears on a table.
That's a great way to think about it.
I mean, you have all the pieces in front of you, but the clock is never going to tick again.
Yeah.
So you get your barcode UMI data, you get your perfect snapshot of that exact millisecond, but the biological timeline is permanently destroyed.
And the clinical implications of that are just severe. Biology, and especially disease, is inherently dynamic.
Give me an example of that.
Well, let's look at how cancer develops resistance to chemotherapy.
Okay.
That resistance isn't a single event. It's not a switch that gets flipped. It is a prolonged, agonizing process of adaptation.
Right, it takes time.
But if you have to kill the cancer cell at hour 1, just to read its mRNA and see what its initial reaction to the drug is.
Then you can never look at that exact same cell again at hour 10.
Exactly. You can never see how it mutated to survive. You are forced to look at a completely different cell in the dish and just cross your fingers and hope it is acting the same way as the first one.
So we are missing the cause and effect entirely. We see the car midair, going back to the movie analogy, but we completely miss the high-speed chase that led up to it. And we miss the explosive crash that happens 3 seconds later.
You're completely blind to the sequence of events.
So for everyone listening, let's just do a quick bulleted recap of where we are. Point 1, cells use mRNA to send instructions. Point 2, to read those instructions with UMIs, scientists have to kill the cell. Point 3, dead cells don't have a future, so we lose the timeline.
A perfect summary.
Which leads us to the big question. How do we put a wiretap inside a living cell to constantly monitor its mRNA without bursting the membrane and killing it?
And this brings us to the massive breakthrough.
Yes. Dr. Plummer's 2026 article introducing TimeVault. They call it a quote-unquote living single-cell technology.
And the timeline graphic in the source material is amazing. They show a cell traveling along a curve.
Right, through points 1, 2, 3, 4, 5, and 6.
So instead of a single dot on a page, that dead snapshot, you have to imagine a continuous waving timeline spanning across hours or days. You are watching the exact same cell evolve over time.
But how on earth do they pull that off without destroying the cell? I went down a serious rabbit hole on the mechanics here. And it all hinges on hijacking something called a Volt particle.
Volt particles are fascinating.
They really are. And they aren't some synthetic nanobot we built in a lab from scratch.
No, not at all.
They are naturally occurring structures already floating inside the cells of most eukaryotes, including humans, right?
Exactly. Nature already provided the perfect microscopic architecture for us to use.
They look like tiny pentagons in the diagram.
Yeah, and structurally, Volt particles are basically barrel-shaped protein shells. And by molecular standards, they are absolutely massive.
It's huge.
For a very long time, scientists were debating their exact biological purpose, but because they are essentially large, hollow, naturally occurring shipping containers, bioengineers realized something incredible.
That they could weaponize them for data storage.
Precisely.
They took nature's empty cargo containers and genetically engineered them into a biological recording device, a molecular flash drive.
It is so clever.
So let's break down the specific anatomy of these time vaults, because the source diagram gets really detailed here. There are 3 internal components we need to understand.
Right. Let's start with the physical casing.
Okay, so first you have the MVP, the major vault protein.
The MVP is the structural wall. It's the physical plastic casing of our microscopic flash drive.
But an empty casing doesn't do anything on its own.
No, it's just a hollow barrel.
You need a way to pull the cell's mRNA inside of it. So researchers attached a chemical hook to the inside of that MVP wall. And this hook is called PABP.
PABP, which stands for poly-A binding protein.
Okay, let's pause and really expand on this, because this is where the biological engineering gets incredibly elegant. Why use PABP?
Well, every single mRNA molecule natively possesses a long tail made of adenine nucleotides.
The poly-A tail.
Yes. In nature, this tail acts as a buffer to protect the mRNA from degrading too quickly while it floats around the cell doing its job.
It's like the plastic aglet at the end of a shoelace that keeps it from fraying.
Exactly. And PABP, that protein hook attached inside the vault, has a natural magnetic affinity for that specific tail.
So a great visual memory trick for the listener trying to hold all these pieces together is to use a simple analogy. Think of the poly-A tail like a fuzzy strip of Velcro attached to every single piece of mRNA.
I love that analogy.
And the PABP inside the vault is the matching Velcro hook.
Right. So by engineering the cell to produce these modified vaults, researchers basically created a trap.
A molecular trap.
As the cell goes about its normal life, just producing mRNA to build proteins, some of those mRNA strands inevitably brush up against the open vaults.
That's smag.
Yep. The PABP Velcro hook grabs the poly-A tail, locking a sample of the mRNA safely inside the vault.
And then the MVP, the major vault protein, acts as the impenetrable shell.
Exactly. Once the mRNA is inside that barrel, it is completely shielded from the cellular enzymes that normally break down old instructions. It is perfectly preserved.
So the cell is actively and continuously backing up its own genetic data into these molecular archives.
Yes.
But wait, here is where I want to push back a little, or at least ask a probing question.
Go for it.
If the vault is pulling the cell's mRNA out of circulation to back it up,
Yeah.
doesn't that starve the cell, the very instructions it needs to survive?
Yeah.
I would imagine grabbing all the blueprints stops the construction project entirely.
That is a brilliant question, and it would if the vaults captured everything.
Oh, they don't.
No. The brilliance of this design is in its sampling rate.
Okay, tell me more about that.
The researchers deliberately calibrate the system so the vaults only capture a tiny fraction of the overall mRNA pool.
Ah, so there's plenty left over.
Exactly. The cell still functions completely normally. Think of a historian standing next to a massive printing press. Every hour, they pull just 3 or 4 newspapers off the line to put into a fireproof safe.
So the city still gets its daily news, the economy keeps running.
Right, but the historian is quietly building a flawless chronological record in the background.
That is mind-blowing. Okay, let's pause and synthesize exactly where this leaves us, because this completely flips the paradigm. Let's do another quick bullet summary for the audience.
Sounds good.
Point 1, we moved away from the old barcode UMI dead snapshot.
Good riddance.
Point 2, we have TimeVault, living cells engineered to produce their own major vault proteins. Point 3, these vaults are outfitted with a PABP Velcro hook that continuously samples the poly-A tail of mRNA.
And point 4, the cell just keeps living for hours or days, leaving a trail of biological breadcrumbs safely archived in these time capsules.
Right, so think about that. We now have a living chronological view of cell dynamics. But how do we know which vault was recorded when?
Because researchers can chemically differentiate the vaults created at different time points.
Oh, wow.
Say, a vault formed at hour 2 has a slightly different signature than a vault formed at hour 20. So they can finally read the full movie of the cell's life, frame by frame, in precise chronological order.
Which opens up entirely new frontiers in how we fight disease.
Yeah.
So let's look at the real-world application of this. I want to go back to that chemotherapy example.
Drug exposure is a huge part of the source material.
Yeah, the diagram specifically has arrows pointing to the timeline labeled time point or drug exposure.
Right. So with TimeVault, you can finally watch the internal machinery of a tumor's survival pivot in real time.
How does that look in practice?
You expose the living cell to a drug at, say, 0.3 on that curve.
Hour 0.
Exactly. And the cell doesn't die instantly. It continues to live and react. Then later, you examine the backup data in the vaults from points 4, 5, and 6.
The subsequent hours.
Yes. And you can see exactly which genes it activates in an absolute panic to pump the drug out of its system.
You are watching its defense mechanisms boot up step by step.
Yes, and this technology captures something the article calls transient states.
Transient states. Let's expand on that term. A transient state is, by definition, something fleeting. It's a temporary biological condition.
Right. If a cancer cell only activates a highly specific mutant survival gene for exactly 5 minutes after being hit with a drug.
The old dead snapshot method almost certainly misses it, because you'd have to get unbelievably lucky and burst the cell at that exact 5-minute window.
You'd blink and miss the most critical piece of the puzzle.
But the TimeVault archives it. It grabs that fleeting newspaper for the safe.
Exactly. It provides an unprecedented map of a cell's molecular weaknesses. Because if you can identify the exact 5-minute transient state a cancer cell enters to survive your primary drug,
then you can engineer a secondary drug specifically designed to block that exact escape route.
Exactly. It is essentially molecular warfare with perfect chronological intelligence gathering.
That gives me chills. And this technology also unlocks something the paper calls lineage mapping. In the source graphic, it's visually represented as a single node splitting into a whole network of different colored nodes.
Lineage mapping is a huge deal for stem cell research.
Right, because if you want to track how a single parent stem cell divides, branches out, and differentiates into a wildly diverse network of specialized daughter cells, like brain cells, skin cells, heart cells, you previously had a massive paradox.
The old measurement paradox.
Yeah, if you killed the stem cell to read the mRNA telling it to become a heart cell, it could never actually divide and become the heart cell.
But now, the parent cell passes its internal archives down to its daughters.
Oh, wait. The filled vaults go with the new cells.
Yes. As the stem cell divides, the filled vaults go right along with it, so you can track the continuous, uninterrupted chronological sequence of gene expression from the original parent all the way down to the final specialized tissue.
That is just incredible. But, okay, this introduces a massive logistical hurdle.
I think I know where you're going with this.
I was thinking about the physics of actually reading these vaults. If I have a petri dish with 1 million cells in it, and they are all dividing and reacting to a drug and they're full of vaults, eventually you still have to collect all these microscopic time faults to sequence the data, right?
Eventually, yes, you do have to read them.
So how do I know which vault came from which specific cell? Wouldn't it just be a massive, mixed-up soup of millions of vaults?
It would be a complete mess.
So to understand a single cell's timeline, you have to completely isolate it from all the others.
And that isolation is the final, crucial piece of the puzzle. And the Spotlight article references foundational research published in Science magazine back in 2025 by Mazzelis and colleagues to solve this.
Mazzelis et al. Let's dig into that.
They pioneered a technique for performing multi-step genomics on single cells inside sub-nanoliter capsules.
Sub-nanoliter capsules, let's do the math on that. A nanoliter is 1/1000th of a liter. So a sub-nanoliter capsule is an impossibly small environment. I looked into how they actually create these. They use a process called droplet microfluidics.
Which is such cool technology.
It really is. It basically relies on the simple fact that oil and water don't mix.
Like salad dressing.
Exactly. So they shoot an incredibly tiny stream of water containing the cells into a flowing stream of oil. And it pinches off these microscopic water balloons.
Yep, tiny droplets.
And each balloon acts as an isolated sub-nanoliter laboratory, trapping exactly one single cell.
And that isolation is vital, because Mazzelis and their team figured out how to keep single cells alive, growing, and dividing inside these unimaginably tiny oil-suspended droplets while simultaneously feeding them chemicals and analyzing them over time.
Okay, so when you combine that 2025 capsule technology with the 2026 TimeVault technology, the potential becomes absolutely staggering.
It really does.
Let's put those two together for the listener. So if we can isolate a single living cell in a sub-nanoliter water balloon,
Right.
and we let it live for days, while it actively records its own internal reactions to a cancer drug using these PBP hook molecular flash drives.
Then we're basically running millions of individual, hyper-isolated clinical trials all at once.
Wow.
We're no longer guessing how a massive population of cells reacts based on the statistical averages of dead snapshots. We are watching millions of individual living cells make complex life and death decisions in real time.
So think about that next time you hear about a clinical trial. We can map the exact lineage of a stem cell or watch the exact moment a tumor evades the therapy, all without ever stopping the biological clock.
It's a total game changer.
Okay, we have covered a massive amount of ground today. Let's do one final summary of the journey we just took before we wrap up.
Let's do it.
Bullet 1. We started with the fundamental flaw of the barcode UMI snapshot. You have to burst the cell membrane with harsh chemicals to tag the mRNA.
Which kills the cell and completely destroys any chronological data.
Bullet 2. We explored the TimeVault solution, hijacking nature's massive barrel-shaped major vault proteins, engineering them with a PABP Velcro hook to grab the fuzzy poly tail of the mRNA.
And letting the living cell silently build its own continuous genetic archive in the background.
And bullet 3. We looked at how isolating these cells in sub-nanoliter oil and water capsules, thanks to the 2025 science paper, allows us to read these vaults without mixing them up.
Giving us the power to capture fleeting transient states, map complex family trees, and track real-time drug resistance.
So the entire field of cellular biology shifts from a discipline of still photography to a discipline of cinematography.
We can finally press play on the cellular movie.
We can rewind the tape, watch the high-speed chase, and actually understand the plot.
Exactly.
But, before we sign off, there is a really profound implication to all this we haven't even touched on yet.
Oh, what's that?
Well, we've spent this entire deep dive talking about reading the cell's history, right? Archiving it.
Right, getting the chronological timeline.
But if we now possess the technology to engineer major vault proteins that can safely store and interact with the cell's internal mRNA over long periods of time. I have to throw this question out to you and to the audience.
Okay, lay it on me.
How long will it be until we use these exact same vaults, not just to read a cell's timeline, but to rewrite it entirely?
Oh, wow.
What happens when we preload the vault with our own synthetic mRNA instructions and engineer it to release them into the cell on a specific time delay?
Like a molecular Trojan horse?
Yes, a molecular time machine that doesn't just passively observe the past, but actively dictates the future of the living cell.
That is a terrifying and incredibly exciting thought to leave on.
It really makes you wonder what the 2030 version of this technology is going to look like. Well, thank you so much for joining us on this deep dive. Keep questioning the world around you. Keep looking for the movie instead of just the snapshot, and we will catch you next time.